I wanted to explain to you what I do. I'm a clinical academic, so I'm a medic, but I actually work for the university and largely do research, although I do see quite a lot of patients as part of my research. And I was going to talk to you about how you take new cancer drugs into the clinic. And as an overview, where we find drugs, what's different about early phase trials, and I'll explain to you about the phases of trials, although we all heard it during COVID repeatedly, but cancer trials are a wee bit different, and then talk a bit about the academic drug discovery we do in Newcastle. So I'm an oncologist, and oncology is the treatment of cancer, and in the UK, that's the solid tumours. Hematologists treat the liquid tumours, the blood tumours, leukemias and things. We only treat solid tumours. And you've got oncological surgeons, you've got clinical or radiation oncologists who use radiotherapy, X-rays to treat tumours, and then you've got the medical oncologists who use drugs. They're all different medical disciplines. We all do medical degrees, and then we train through different royal colleges to get accredited as oncologist. So I'm a medical oncologist employed by the university and an honorary consultant at the Newcastle Hospital. And my job is to run a cancer trials unit. And when you think about cancer, I mean, I hate the battle about cancer, but you can think about the treatment as the three forces. And that's because, as an analogy, the surgeons are the senior service, like the Navy, because they've, for hundreds of years, people have tried to treat tumours, particularly lumps and things you could see on the limbs, on the legs or arms, with surgery, and that's an amputation set from the 1800s. Radiotherapy was discovered by Röntgen in 1896, and within months, it was first used within months the next year for treating tumours. That's the picture on the left is a radiotherapy kit from the early 1920s, no protection for the poor nurse operating the radiotherapy there. And people giving radiotherapy used to get radiation-induced tumours. The other picture is the second X-ray ever taken, and that's Röntgen's wife's left hand, because you can see she hasn't got a tumour. You can actually see her wedding ring, the metal of her wedding ring. He took a picture of the door, and then he took a picture of his wife's hand while he was working out what these weird X-rays were. But the other thing, 50% of patients get radiotherapy, so it's worth thinking of them like the army or the cavalry, because about half of our patients at some point in their journey would have radiotherapy. But the other thing that's with medical oncology, the junior service, because there aren't as many medical oncologists, there only have been cancer drugs around since the 1940s. And the first one was actually developed, it's a nitrosourea, out of the mustard gases, the nitrogen mustards used in the First World War. And there were stocks of mustard gases and accidents with them in the Second World War as well. But it was noticed that people who survived being gassed dropped their blood counts. And in the late 1940s, early 1950s, the first trials were done treating leukemias with nitrosoureas, one of the compounds that's in the nitrogen mustard gases. So that's when the cancer drugs started. And actually, we use a lot of poisons, and we use a lot of natural poisons. There's a periwinkle there, the pink flower, the vinca alkaloids used in childhood leukemias. Periwinkles are pretty poisonous. The American oak apple gave us etoposide. You know, we work with a company that looks at the natural poisons you get out of animals in the sea. If you think about a jellyfish sting, those are really quite poisonous compounds. You get blisters because the skin cells get killed. So lots of people have looked at natural things, made them into poisons, and then tried to give them to patients. What we now try and do as well is model and make drugs to fit or block what we want to block. But that's where we find the new drugs. And it can all take a very long time. And this is about 10 years old now, this slide, showing, because things are changing, thankfully, that you can go from thousands of compounds through to just one eventually licensed. But actually, what I wanted you to focus on was the clinical phases here, because it, again, can take us 6 years. And I do phase 1 and phase 2 studies of clinical development, working out how do you safely give a drug. And this is really frustrating for the people developing drugs, but also patients waiting for new treatments. But you do need, for safety reasons, to do early phase clinical trials. And cancer trials are different from all other parts of medicine. So in all bits of medicine, a phase 1 study is the safety study, working out the dose, trying to understand the toxicity, and to recommend what you call the phase 2 dose, the dose that would go ahead. In every other medical discipline, those studies are done in healthy volunteers. And the phase 2 study is the first time patients take a drug, a new drug. But if you think what a cancer doctor wants to do, we've got cells within a patient that have gone catastrophically wrong, but they are still that patient's cells. You want to target those cells specifically, and to treat them, you actually want to kill them. So if you can't cut them out like a surgeon, if you're a doctor who gives drugs, you want to kill them, the cells. And you can't really ask healthy volunteers to take those sort of drugs, even small doses. So we have always done, and the patients I was seeing in clinic this morning have all got tumours, all know they've run out of standard treatment options, and are therefore prepared to take something experimental and want to explore that as an option. So those are phase 1 studies. Phase 2 studies, you've got your dose and you're trying to work out, does it work well enough? And then the phase 3, the late phase study, is where it's randomized, the randomized control trial that we were all waiting to read out with the COVID vaccines. Those are the late phase studies, and then a drug can go to license. So we do phase 1 and phase 2 studies. And traditionally, when we just used those poisons I talked about, we had this design where we treated 3 patients with what we hoped was a safe but not too low a dose. And if they didn't get bad side effects, we often would double the dose and treat another 3 patients. And if one patient got bad side effects, you'd treat a few more. And if it was 1 out of 6, you'd go up a bit more cautiously, not the doubling still, but you would keep going up. But if it's more than that, you would say, well, that's too much, and you'd go back. And all chemotherapy has traditionally been, we treat patients at what we call the maximum tolerated dose. The biggest dose we think we can get away with, because you want to hit the cancer hard, and then you want to give the patient a break to let the rest of them recover, and then you go back in again. So that's how we'd always done a trial. But in the early 2000s, a couple of really great, and this is actually a very good paper to read, Hanahan and Weinberg, a couple of really great researchers wrote a paper about the hallmarks of cancer, all those things that went wrong in cells. Now, they updated it in 2011, and initially there were 8 hallmarks, and now they said there are 10 things that commonly go wrong. But what was really encouraging is by that stage, other researchers have thought, well, if that has gone wrong, can we find out in a cancer cell in a dish, understand that, and then can we target it? And now right round that circle, we have drugs, a lot of them licensed, but certainly drugs in the clinic targeting all the hallmarks of cancer. Now, this is not just poisoning with chemotherapy. So we're seeing very different, we're needing to do our trials in a different way, because then you want to find the right biological dose. You don't just necessarily need to poison as much as you can get away with. So it's changed, but it also means we see slightly different side effects. So we've had to understand a lot of, over the last few years, it's been very interesting, but we've had to understand a lot more things. And that trial design, I mean, from what I grew up with, 3 + 3, I could do the maths on that. We didn't need statisticians. We now have really complicated trials, which are much more fun, much more interesting, and more likely to help patients with these complex designs where within one study, we can work out various doses, which is what this is showing, and combinations. And when you're doing a combination of 2 new drugs, you could go up with one and down with the other. So you can start splitting that. So we do lots, and we have much, talking with the regulators, much more flexible trial designs. So those of us who do safety studies get the maximum possible we can out of those studies and try and keep, work out the safe and the right doses to take drugs forward. The other thing that's changed is that ability for us to be able to sequence. And the fact, you'll have read about the 100,000 Genome Project, but also the Human Genome Project, firstly, was billions of dollars to sequence one human genome. We can now do that for £1,000 or under, and that is changing how we do our trials once again. And it's also changing our trial design. And this is a picture partly to show you something much beloved of oncologists, what we call waterfall plots and swimmer plots. And it does sort of make sense. So larotrectinib is a drug that targets a rare thing that goes wrong in some tumor cells, and it blocks it. So we end up with tumor drivers, a protein that's gone wrong that's just driving the cell to grow. If you can, it's like having a light switch stuck on. If you can switch it off again, you can block that. And the cancer cell is often very dependent on that. And if it gets blocked, it often dies off. This is a rare mutation, but they designed a study. They did a basket study. They said, it doesn't matter where the tumor came from. And this study took patients from age of 1 up to 90 something as well, because as long as the tumor had the mutation, that was the basket. You have the mutation, you're in the basket, you can have the drug. And the waterfall plot is the patients on the waterfall going downwards, and these are each individual patients on the study, means their tumor has shrunk. So you measure it when you start, and that's 0.0. If the tumor sadly grows, the line goes up, and if it shrinks, the waterfall goes down. So that shows how many patients responded because our studies tend to be small. The swimmer plot, this is months along the bottom here, is how long the patient stays sort of swimming along in the lane on the drug. So that shows how long patients on average were staying on the drug. So that's a basket study, which we can now do if we can understand about the tumor. The other sort of studies we do, and there's a big run out of Birmingham National Lung Matrix study, sponsored by and paid for by, funded by CRU, Cancer Research UK. This said, well, it's lung cancer, but if we profile, and we started off looking at 28 different genes that we might be able to target, and it got expanded. And all of the patients were under the umbrella because they all had lung cancer, but the different spokes of the umbrella were different drugs depending on their tumor mutation. And again, we saw lots of responses because we were doing this personalized medicine. And this is changing those really depressing timelines. And this is just an example of a lung cancer drug called crizotinib, which was being developed, and it was going into the clinic. But it was discovered in 2007 that some lung cancer patients have one of these abnormal proteins, which means that they are sensitive to this ALK inhibitor, because that's the bit that's stuck, switched on, and is driving them to grow. So 2 years after that was discovered, on the phase 1 study, patients with this ALK inhibitor, this ALK positive lung cancer, were responding. 2 years later, it was so clear this worked, the drug was approved in that group of patients. It's only 4% of lung cancer. It's rare, but it's a drug that works. So that's the other thing that we are seeing change with, using the science and evolving what we do. And what we do, and what I recruited 2 patients to this morning, now with all of the patients referred to the trials unit, we offer them something called Target National. And this is a blood test where we can look at 360 potential tumor genes that might be in their blood. And the reason you can do that is that all of us in our blood have little fragments of DNA circulating, because as our cells naturally turn over, the DNA gets broken down, but some of it comes out in the blood. If you've got a tumor on board, you can find the normal DNA and you can find the tumor DNA. And it takes us a couple of weeks to get a report. We send the blood off, and it says that the tumor fraction in this patient's blood, it can be 20%, but mostly it's down in the fractions of a percent. 1% is tumor DNA, but this and this has been seen. And that's how we're now matching patients to trials. We're part of the Experimental Cancer Medicine Network, funded part by the government and part by CRUK. And we've linked up all of our trials. And if we get patients where we found a mutation, we will then say to them, we haven't got a trial, but there's one in Manchester or one somewhere else. And we're also setting up a study called Determine, which is another of these umbrella studies, where if you've got a rare mutation, there's various arms opening up, where you can then, patients include across all age groups can access drugs, again, to try and look for and understand who's most likely to respond to the new drugs we've got. So that's, that's how we find new cancer drugs, but it's also how we try, because we've got patients who are, at the end of a patient journey, but saying, I'd like a trial, and I understand all the risks and all the things you don't know. So we really do try to match them as well as we can to drugs. But Newcastle University, alongside the Institute of Cancer Research in London with the Royal Marsden Hospital, are the two places in the UK that do academic cancer drug discovery and actually have taken drugs through, and they then eventually become licensed and in the UK NICE approved and available for treatment. And there's been 40 years of active cancer research at Newcastle University, starting in 1982 with a cancer research unit. In 1990, what was Cancer Research Campaign, the forerunner of CRUK, funded the second academic drug discovery unit up in Newcastle. There was one in London, as I said. And then through the years, we've become, as I said, an experimental cancer medicine centre. Then in 2009, we became a CRUK Centre of Excellence for doing drug discovery and the trials we do. 2009, we also got a nice new hospital at the Freeman. And the next year, the Great North Children's Hospital opened, because we do both children's cancer trials and adult cancer trials. So this research has been ongoing for many years in Newcastle. And as I said, we are a CRUK centre. There are 7 CRUK centres now, 4 major centres and 3 centres. There's 4 institutes as well, but there's the 17 ECMCs, there's clinical trials units. So that right around the country, there's this structure to try and find new cancer drugs and do trials. And we've got quite a nice little geographical space up in Newcastle. And I was going to show you that. So we recruit patients from up to the Scottish border, although I can actually recruit across the Scottish border, and we talk a lot to Glasgow and Edinburgh, our colleagues up there, if people are on the border, and we'll swap patients. And then down into North Yorkshire with Leeds and Manchester. Manchester, Newcastle and Glasgow with the Marsden in London are the 4 largest early phase trial centres in the UK. So bizarrely, we've got a big triangle of that activity up in the north of England. When I'm talking in London, I always point out that we've got all the nice bits. I have to say England, all my Scottish friends get cross because they say they've got beautiful bits of Scotland. You'll see I haven't, when I do my slides, been able to take away my lovely picture of the tree at Sycamore Gap, but maybe I have to alter that one as time goes by. But so that's the area we do, and Newcastle is fantastically set up to do this. So not just with the hospitals, and we now do all the treatment. We've got the Northern Centre for Cancer Care in Cumbria, which is over in Carlisle. We now treat all the patients in Cumbria as well for normal cancer treatments as well as trials. But we've got some great facilities in Newcastle as well to support us and do that research. And this is a picture. I'm assuming you all know Newcastle well. So we're over at the Freeman. There's Gray's Monument. There's the football stadium, which I don't need to label for you all. But there's actually all these bits and pieces. The Great North Children's Hospital, the science building where I was yesterday talking to colleagues doing over Paul O'Gorman. There's the Wolfson building, which is just children's cancer research. And all of those is sort of pulled together so that we can deliver cancer research and new treatments into the clinic in Newcastle. And I do this, I'm the clinical end here, as the the girl at the clinic. It used to be a picture of a boy, but I changed it to a girl. And Steve Wedge is my co-lead of the CRUK Centre, and he's the bioscientist. So what the scientists do is they look at cancer cells in addition, say, right, what's gone wrong? What can we target? They find that protein. We have structural biologists who then model that, and they call it in silico. They make the drugs initially on the computer, and then the chemists come in and help and make them better drugs. You then have to, what they do, hit to lead and lead optimization, which is basically a lot of it is making the drug soluble, because any drug you give to humans has to dissolve in water because of the way we're made up. You then do need to understand something about toxicity. And I'm honest with my patients that no drug goes into the clinic that hasn't had some animal testing as well. It used to be just rodents in Europe. In the States, it had to be a higher order species as well. But the rules are closer together now. But there is some, as limited as they can get away with, but there has to be some evidence of what you might see with side effects in animals before you can take a cancer drug into humans. But then we do the human trials, and that's what we do. And it is a bit of a long and winding road. It's another picture of the Lake District with our daughter. I was not in charge this day, and she's definitely wandering off the path, which we feel happens. But it's not an easy way. It's not an easy path, but it can work. And there are 2 drugs now. So Newcastle actually is the only place that's done 2 drugs all the way through. The Marsden's done 1, but we've actually done 2 drugs that are now licensed and NICE approved, rucaparib and balversa erdafitinib. And I've been very involved with the development of rucaparib. And I was going to tell you that to finish up as a story. And it's a PARP inhibitor. And PARP stands for poly-ADP polymerase. And PARP is a very cool enzyme. And we all have, in all of our nuclei, we have 1 PARP molecule per kilobase of DNA. And it's a little enzyme, and it's inactive. But it's got a thing called a zinc finger that if there's a nick in the DNA, it binds to it. It switches the enzyme on, and it makes a polymer of ADP ribose, which is negatively charged. So if you've got a PARP molecule either side of a break in the DNA, and each one puts a negative charge on itself, it flexes the DNA apart because negative charges repel chemically, and then other things come in and repair the DNA, and it protects our genome. And so that's what PARP's supposed to do. And when you give chemotherapy, you damage the DNA, and the cell tries to repair it. So Newcastle was trying to make a drug to improve chemotherapy. And they made a PARP inhibitor, and it was ready to go into the clinic in 2003. And that's when I met Peter in my clinic. He at the time was 43, a painter and decorator. He had teenage children. He had an inoperable tumor, which you can see outlined yellow here on his CT scan of his abdomen. And he had no treatment options left. So he wanted to talk about a trial, and he knew his only other option was symptom control. So when I talked to him about what we did and didn't know, he said he was prepared to take a drug where we didn't know the dose, we didn't know the side effects, we didn't know if it would work. And if it didn't help him, it might help somebody else. So that's the sort of patients that I see in clinic. Now, the story of PARP had started a few years before with a lady called Barbara Durkatz, who at that 1990 was working in Newcastle. But a decade earlier, working down in Brighton, had discovered this enzyme reaction. And she showed that if you were killing cells with chemotherapy and blocked this enzyme reaction with a weak PARP inhibitor called 3-aminobenzamide, you got more bang for your buck. You killed more cells with a given dose of chemotherapy. So she said, this might be the way to make chemotherapy work better. And when CRUK invested in Newcastle to do drug discovery, it was our first project. So the first PARP inhibitor, this is Barbara's PARP inhibitor. And the reason for showing you this, I talked about things binding in pockets. It's the nicotinamide bit that binds in the PARP enzyme and sets off this whole enzyme reaction. Can you see how that's roughly the same shape? But it binds and it sticks. And what the Newcastle chemists started doing is they kept that shape, but they added bits on to make it bind harder and stronger and stick and block the enzyme. And then they had to add the bits to make it soluble in water. And you can see these first compounds from Newcastle University, or NU compounds. Then, to do cleverer chemistry and get it really right, they partnered with a small company called Agaron in the States, so they became AG compounds. And it was AG 14447 that we took into the clinic when CRUK sponsored and ran a trial for us. And as I said, we were trying to make chemotherapy work better. And our PARP inhibitor, this is chemotherapy killing cancer cells better with the PARP inhibitor in a dish, or actually human tumors growing in an animal, and you could start to cure them in the animal by giving the dose of chemotherapy with the PARP inhibitor, so you could make your chemotherapy work better. So that's what we wanted to do. And so we started giving our PARP inhibitor with chemotherapy, and Peter was the second person in the world ever to get a PARP inhibitor. There are now 4 licensed PARP inhibitors used to treat cancer patients as a really powerful class of drugs. But it all actually started off in cancer medicine in Newcastle. We showed in that first study, some of the patients very generously allowed us to take a little piece of their tumor and show that the drug was getting into the tumor and blocking the enzyme in the tumor. We knew we were blocking it in the blood from blood tests. So you can see here, this is a low dose where it was only 80% blocked. As we took the dose of the PARP inhibitor up, we blocked it more strongly in the tumor. Peter, his tumor started to shrink. And this is after a year of treatment. It was getting smaller. After nearly 2 years of treatment, I was getting a bit nervous. He was still doing well, but he'd had quite a lot of the chemotherapy drug by then, and I was starting to worry that we were using too much of that. So he then went on to have further surgery because this tumor was now considered operable. But at around that time, the scientists in Newcastle and also down at the Marsden brought out some really interesting data. So the science marched on while we were in the clinic working out how to do dose this drug safely. That if you had an inherited BRCA defect, which is the thing that commonly causes in families breast and ovarian cancer, the PARP inhibitor might just work on its own. We didn't need the chemotherapy. And the Newcastle team had told me this data was coming out. So we went back to CRUK and said, you know, we've done this study. Well, we've got a really good idea for the next study. We actually ought to just do the drug on its own in this particular group of patients. And that was my first PhD student did her project around this. We set this study up, and the second patient we treated responded. Now, we became very topical at the time because Angelina Jolie said that she had a BRCA2 defect. Pierce Brosnan's wife had died of ovarian cancer, and his daughter has the defect as well. So there was a lot of stuff in the press around this as well. We had said this wouldn't work. 2 years before, when I went to the ethics committee to say we've got this completely new class of drugs, but we need to give it with chemotherapy because it doesn't kill cells on its own. And it doesn't usually, but we were wrong. If you get the genetics, the driver mutation right, it'll kill cells on its own. And this was the key paper where here is, you can increase the dose of the PARP inhibitor and the cells, 100% of them pretty much just remain alive. They don't care. If they've got a BRCA defect, they die. If you then transfect, genetically put the BRCA back in, they stop dying. So it was clearly that you needed the two things together, a thing called synthetic lethality, where you've got one genetic defect and you cause another problem, and that's synthetically lethal. And the reason it worked, and this was very cleverly worked out in theory by a scientist down at Sheffield University, is that all of us every day get little spontaneous DNA strand breaks because they just our cell turnover, you get things called reactive oxygen species. They nick the DNA. Normally, PARP just comes in and repairs it. I said you've got 1 per kilobase of DNA. You've got loads of PARP. It'll just get repaired. If you block PARP, it doesn't really matter if the cell's not being very active. But if it tries to divide, and that's what cancer cells are doing, it's like peeling 2-ply wool, if anybody's a knitter, peeling it apart. The strands will then break and you get a snap in the DNA, which is a really bad thing to happen. Now, we have another DNA repair mechanism to stop that because it's such a bad thing, called double strand break repair. And BRCA1 and BRCA2 are important parts of that. So if you block PARP and the cell tries to divide, if BRCA is there and functioning, the BRCA wild type, genetic word for normal, it'll just repair it. So it doesn't matter. It can keep growing with a PARP inhibitor, which is why it doesn't work on its own. But if the tumor has lost BRCA, which is what happens in patients with the germline BRCA defect, that is lethal and it would work on its own. And that's what happened. Here's another waterfall plot, one of the reasons I explained them. This was Ariel 2, the second study we did with the PARP inhibitor. 80% of patients with BRCA-related ovarian cancer will respond to rucaparib, the PARP inhibitor we developed, because their tumor is really vulnerable to it. So the FDA saw this and fast-tracked the drug to registration. It was the first PARP inhibitor to be given the breakthrough designation. The company, Clovis, who oncology were developing it by then, sent the trials unit, because we did the first patient, a paper, a sort of paperweight plaque thing, which has got the structure of the drug on for us. But CRUK gave us their first ever translational team prize for team science, team working to get this drug through and over the line, the way that the whole team had to pull things together. And the initial license for rucaparib was in BRCA germline associated cancer. So here's the timeline on a clinical development story, and acknowledging that CRUK kept investing, kept supporting us to do studies. The license, it often is drugs are licensed in the States first. It was nearly 2 years later that Europe licensed the drug. So I was asked to organize a celebration event, which we did partly science in the afternoon, but we did it as a public engagement event at the Great North Museum. And what was really special for me, because it was a public engagement event, I still occasionally saw Peter in clinic. And I said to them, they came incognito, would he and his wife like to come along and hear this story from the other side, which he'd been part of? And that was just lovely. He, felt it was the right thing for him to do a trial. We were incredibly lucky that he had the right sort of tumor to respond. And just to wrap this up, where we do all this is the Bobby Robson unit. And so Bobby Robson, he's been very open about this, so I'm not giving away any secrets. He had cancer. He was my patient, and I actually, with the sort of cancer he had, didn't have any good treatment. So he said, well, I'll have a trial then. Why would I not? I'm prepared to try. We were then fundraising for the unit, and I asked him for advice on contacts as is well known. And this is one of the fundraising events. He and his wife decided they would help us fundraise while he was having treatment. And this is a picture up in my office on the unit that he sent me from one of those fundraising events, joking in, why did you ask? And glad you did. And he felt this was a really important thing to do, was to help us raise money. And the foundation has done a phenomenal job and raised millions of pounds for cancer research in the northeast of England. And the unit is named after him. And he opened it in 2009. We've now got increasing number of trials. They're much more complex, as I've shown you. This is Lady Elsie coming to one of our public engagement events to chat to us and look around the unit. And we certainly do medical student and student events to show them what we do on the unit. And we've now grown out of that unit, and I met up with Sir Bobby's son yesterday at another meeting, because we're thinking about what do we do. We've got a waiting list of patients interested in trials. We're now turning down studies, so we're having discussions with the hospital, the trust, the university and the foundation over whether or not we can think about and design and grow a bigger unit so that we can offer more trials to more patients in the Northeast. So I hope I've shown you that, I mean, cancer medicine I think is fascinating, and I guess that I hope that's come over. It's changed enormously in the 20 years I've been a consultant. We now don't just, I think it's a long time since I've prescribed any chemotherapy. We have monoclonal antibodies, we have these small molecule targeted drugs, we have immunotherapy drugs, which I use a lot of, and we actually have cellular therapy. So there's lots of exciting things and change in research. But the first step in the clinic is always about safety. And that for all doctors, the first thing that we all promise to do when we start at medical school is to do no harm. So thank you very much for listening.